Hello everyone and welcome to today's class on biosynthesis of protein with a special reference to the chemical reaction occurring in translation reaction. Let me begin this lecture by asking you a simple question. If I say that I have my eye color similar to that of my father, what is the reason? And if you are towards a biological background, if you are a biologist, then you may say that the chromosomes a child inherits carry genetic information that determines your eye color. The answer will be like that if you are a biologist. That means genetic means DNA, gene, DNA, nucleic acid. And if I show the structure of nucleic acid, it will be like this. And another student, he can answer like this. The protein products like pigments gives eyes their color. Protein means polypeptides, amino acids. The structure may be like this. So one answer is it is genetics, it is the reason of that eye color is DNA. And another is the answer is the reason is a protein which gives the color to the eye. I will say that both answers are correct. So then you may ask, so what's the relation between gene and proteins? So I will show you 2 points. Genes contain the information needed to make functional molecules called proteins. The first statement. And nucleic acids such as DNA or RNA, they control the biosynthesis of proteins. So in order to understand these 2 statements, you have to understand the mechanism of biosynthesis of proteins. Then you will understand these 2 points and what is the relation between gene and proteins and what is the relation between these 2 answers. Okay? So before going to the mechanism of biosynthesis of protein, we should understand about a factory where the protein biosynthesis occurs. I will say ribosome is the most wonderful, amazing factory in the nature where the protein synthesis is occurring with 100% accuracy. This is really an amazing factory, most amazing factory in the nature. Here you can see this is a pictorial representation of ribosome. This is what you see in orange color is ribosome. This is the big ribosomal unit. There is a small ribosomal unit. And the ribosome is made up with ribosomal RNA. And then you can see a messenger RNA. You can see tRNA, transfer RNA. You can see where peptidyl RNA, where peptide, protein, you can see. So this is the structure of ribosome where protein synthesis is occurring. Biosynthesis of protein contains 2 major steps. First one is transcription and the next one is translation. In transcription, the information contained in DNA, the double-stranded DNA, is used to synthesize mRNA. That is the information that I just wrote the information to another book. That is transcription. The information in the DNA is converted to mRNA. That is transcription. In translation, the information in mRNA is converted to protein. That is another language. Protein is another compound. That is polypeptides. RNA is polynucleotides. So the information in the polynucleotide, mRNA, is converted to another compound, another type of compound, protein. That is known as translation. So these are the 2 steps in biosynthesis of protein. So I will explain how transcription occurs. You see, DNA in the first step, the DNA double helix unwinds. Now you have two strands, and one strand is the template strand. And in template strand, you can see TACT, and the monomers of the RNA will align. For example, opposite to T, A will align, opposite to A, U will be aligning, and opposite to C, G will be aligning. That is base pair rule. So according to the template strand, the monomers A, U, G, etc. are aligned. And finally, a chemical bond is formed between them to form the RNA. So this RNA is formed according to the information contained in the template strand. So this is transcription. Okay. Now, in translation, this is the real peptide bond. This is where the real peptide bond is formed. In translation, you see mRNA is there in the ribosome. This is the pictorial representation. I will show the chemical representation later. You see when a start codon AUG comes, the protein synthesis is initiated. Now AUG is the codon, an amino acyl tRNA with anticodon UAC comes. When the start codon AUG comes, UAC comes because A is complementary to U is complementary to A, base pair rule, and G is complementary to C. So UAC binds to AUG perfectly, so that this amino acyl tRNA, that is this tRNA carrying this specific amino acid, binds to this site, so that is ready, that is inside the ribosome. So the first amino acid is always methionine derivative. It's a methionine amino acid. Okay, and then next is CAC, the anticodon which will bind to CAC will be GUC. So only GUC anticodon will be binding with CAC. So only this amino acyl tRNA or this tRNA will be binding here, which is carrying histidine. So the second amino acid is inside the ribosome. Now, see, this methionine is transferred to histidine. So the dipeptide is formed. That is known as peptidyl transfer reaction. I will show the chemical reaction in the next slide. Okay, now it's dipeptide. Similarly, different amino acids will be coming to ribosome according to this codon-anticodon interaction. For example, here ASP is coming, GAC opposed to that CUG will be coming, and that will be that amino acyl tRNA will be binding to this side. So ASP is inside ribosome, and this peptide is transferred to this place. And then finally a code UA, that is known as a stop codon. When that stop codon comes, what happens? The peptide is cleaved, and that's a signal for stopping the peptide synthesis. So the peptide is released, the polypeptide is released. So the protein, according to the information contained in this mRNA, is released. This is how protein biosynthesis is occurring in ribosome. I will show it here. Okay. Here, this is the chemical step. Suppose, so this is already some peptide is formed here. So the next amino acyl tRNA, the code is UCG. UCG means the only amino acyl tRNA which will be coming is with anticodon AGC. So if the anticodon is AGC is coming, that will bind to this side. That is known as, then that amino acyl tRNA will be accommodated. That tRNA will be in the ribosome. No other tRNA will be accommodated in the ribosome. So that is known as accommodation. And after it is accommodated, you see this amino group is in a perfect position to attack this carbonyl molecule. So that will attack the carbonyl molecule. So what happens? This bond is shifted to oxygen. So this bond is cleaved. So this new peptide bond is formed, a new amide bond is formed. So this peptide is transferred to this molecule, this amino acyl tRNA. So this is known as peptidyl transfer reaction. You see, this amino group attacks here. So a new bond is formed here. So this tRNA leaves. So this amino acid is also included here. A new amino acid is incorporated in the peptide. I will show it in a next slide. You see here, this is another pictorial representation. After this is mRNA. If codon anticodon interaction is okay, this amino acid tRNA is accommodated here. Now this amino group is in a perfect position to attack this carbonyl molecule. It attacks here. This goes here. So this leaves. This tRNA leaves. See, this peptide is transferred to here. So this amino acid is incorporated into this peptide. So new amino acid is added. Similarly, you see, this reaction continues. So that a full-length protein is formed according to the information contained in the mRNA. This is how biosynthesis of protein occurs in ribosome. So this is the chemical structure showing the peptidyl transfer reaction. This is the amino group of the amino acyl tRNA. This amino group, the lone pair of nitrogen, attacks this carbonyl molecule so that this bond goes to here. Here the new peptide bond is formed here. This amino acid is incorporated into this peptide. So this is the chemistry of peptidyl transfer reaction occurring in the ribosome. So finally, the stop codon comes and the release factor comes. The peptide synthesis, the ribosome peptide synthesis stops and the peptide is released. The free polypeptide is released and the peptide synthesis is finished. So we get a full-length protein according to the information contained in the mRNA. Okay, thank you very much.